Well, it's time now. Good afternoon to everyone here. I'd like to thank you by the presence here in our fourth Symposium in Comparative Biology. I know that's kind of weirder to talk in English with Brazilians, a Brazilian talking English with Brazilians, but Brazilians and Colombians and Venezuelans and other people. But I think it's a good opportunity to see in person how a professor in USP has a terrible English like me, for example. Okay? A joke. No, just a joke. No? So, this is the fourth Symposium Comparative Biology carried out by our postgraduate program in Comparative Biology that is based in our school, the Faculty of Philosophy, Science and Letters at Ribeirão Preto, in the University of São Paulo. So this activity has been conducted since 2016 with the aim of being a facilitator of international exchanges, helping to improve communication in English, since all the activities are conducted in this language. Also, it's an opportunity to show to us, both students and advisors, what kind of research is made in our program. Thus, this event provides a meeting point for our students, advisors, who can thus exchange experience in their research and interact with their colleagues. I have some photos here from our story. The organization of the meeting is conducted by me, Professor Milton Grupo, and our Vice Coordinator, Tomás Ferreira Domingues, and of course, our excellent Secretary, Vera Cássia Cicilline de Luca. The first symposium and the first 82nd symposium were held in person, like this in 2016. This is the Lucienne Lison of class, and were given by the advisors of the program, by our postdocs, and by students who have done internship abroad. And the second was also in person, okay? It's in 2018. Some fresh faces here, we can see some of the faces are still here. But in 2021, we had a virtual meeting, totally virtual meeting, fully online. And in this occasion, despite the limited human contact, the virtual format allowed us to invite researchers from other countries, and they were able to offer to us lectures in their fields, much enriching the event. And as I told in that occasion, that edition also served as a way for students who were more isolated in their homes, like us, all of us, to interact in an event that brought together all our postgraduate students at a time when the pandemic seemed to last forever, and when everyone needed more intense emotional and psychological support. So this edition here will be the first in-person symposium after the pandemic. So this year the event will count with 3 researchers from our program: Professor Wilfried Klein, that's here in some place here, there, and Professor Liran Alchoer Pereira Martins, both from our program, who will present their lectures on the main lines of research that we currently have in our program. And I would like so much to thank Professor Wilfried and Professor Liran, because all the time that we call them to participate, they always are willing to help. Thank you, Wilfried and Liran, that is not here. Tomorrow, we will come with a lecture given by Dr. Linnea Hess from the University of Hamburg, Germany, who will talk on new techniques to obtain 3D images in biological studies. It will be very interesting, I think. Professor Tomás Ferreira Domingues is in his way. Currently our vice coordinator and the next coordinator of the program will give us in a few moments a glimpse about the program and future perspectives to us. In addition to these lectures given by our guests, there will be 2 sections of lectures, both given by students that took part in activities of internship abroad. Professor Thomas here, welcome. These talks are important to show the research they've done, and they have been a stimulus to other students do the same. That is, to do internship programs. There's also a lecture that will be given by Dr. Wafa Halabi, that's here. Wafa, that is currently a postdoc at the Max Langer's laboratory, and she will talk about paleontology. Is that correct, Wafa? Of course. Finally, this year the symposium features a poster section that is here. This is the photo of the second symposium that was in 2018 in the Invicta Brewery. Okay? This section of the posters will be an opportunity to present the results of the students and talk a little about science in an informal place with a good beer, as in Invicta they have. There will be also a popular balloting, and prizes will be announced tomorrow for the best posters at the end of the event. So tomorrow, after midday, we have the announcement of the winners of the prizes, okay, in the posters. I would like to thank our organizing committee, our vice coordinator, Professor Tomás Domingues, that is here, André Esguícero, for the book of abstracts. Everybody has received the abstract book, okay? That's okay. If you notice errors or some mistakes in the book, please contact us, because we have to obtain the registration of the book to be registered. And would like to thank Vera Cassa Cecilino de Luca. And I have a personal request. I would like to all of us to send in good thoughts and prayers to Professor John McNamara. I don't know if you know, but Professor John is in the hospital and treating his health. Professor John is very important to our program because he's one of the founders of the program, together with Professor Fernando Mantelato, that is in the final role there. And Professor John was and has been extremely important to consolidation of the program, and have been for many years the coordinator of the program at the very beginning of it. Okay? So if you could send good thoughts to Professor John, and I would like to send him the best in the rapid recovery as soon as possible. Now I would like to turn the floor and invite some friends of mine. Professor Daniel Junqueira Dorta, that is here, from the Department of Chemistry from our faculty, and Professor Daniel is Vice Coordinator of the Postgraduate Commission of our school, and we have worked together in the commission called CPG. And I would like to invite also Anaís Freitas Silveira, that is the students' representation of our program, to compound this table, and also Professor Tomás Ferreira Domingues, that soon will be enlighten us with his lecture about the future of our program and future perspectives. Okay? Thank you very much, Daniel, Tomás, and Anaís. Some few words. You can use both this microphone or this one, okay? Daniel also, first one. Professor Daniel.
So, good evening, everyone. First of all, I would like to thank you, Professor Newton, and all the coordination commission of the Comparative Biology program to this invitation. It's an honor to me to be here representing the, how can I say, the Pós-Graduação committee or commission, I'm not sure. Here from the Faculty of Philosophy, Science and Letters at Departamento. It's a great honor to be here at the opening ceremony of this 4th Symposium in Comparative Biology here at the University of São Paulo. This gathering marks a significant moment in our academic calendar where minds covered, ideas flourish, and the pursuit of knowledge takes center stage. The University of São Paulo has been a bastion of intellectual curiosity, and today we find ourselves in the heart of the Faculty of Philosophy, Science and Letters, a unity within our esteemed institution that stands as a testament of our commitment to diverse areas of knowledge. I'm particularly pleasant to say a few words about the Postgraduate Committee of this faculty. This unit holds a unique position within our academic landscape, boasting 10 postgraduate programs that span across a spectrum of disciplines from the humanities to the biology and exact sciences. This unit exemplifies our dedication to fostering interdisciplinary research and innovation. And this program represents not only the breadth of our academic pursuits, but also the depth of our commitment to advancing knowledge in a collaborative manner. It's through initiatives like this that the University of São Paulo continues to be a trailblazer in the higher education, publishing pushing boundaries and understanding discoveries. As you embark on this symposium, in comparative biology, let's celebrate the spirit of inquiry, collaboration, and the pursuit of excellence. May the discussions that unfold here today contribute to the scientific exploration and deepen our collective understanding of the biological world. And let's remember that the unit has infrastructure that makes possible, highlights the potential of all of our PPGs and of our postgraduate programs to contribute to the advances in scientific, technological, and innovation research. So it's a pleasure to be here, and I would like to finish with my near and fast talk, congratulating the Professor Milton and the program to be able to get all these students here and make a symposium like that. It's, as Professor Milton said in the beginning, it's a nice experience, and to be here and talk in English, you see that I may try to read these words that we try to say without reading, but it's a very nice experience, and you will be in the future to help you be the front, talking to the others and explaining your researchers and making contacts, and it's a great moment, and let's I forget the word, but let's make the best of we can from the opportunity to have here. Okay, so thank you again for this invitation, and congratulations very well.
Thank you, Professor Daniel. Professor Daniel is very important because he is from the CPG committee. I don't know if it's a commission or committee, I don't know the official words, but it's important the presence of Professor Daniel here, because we have a commission that is ahead of all our 10 programs in postgraduate program here in our faculty. So we are one of the 10 programs here in our faculty, and Professor Daniel is the vice coordinator of this very important committee. Thank you very much, Professor Daniel, for being here. And Anais, could you bring us a few words, please? And after Professor Tomás.
Good afternoon, everybody. I actually just wrote some short lines that I'm gonna read. So, for those who doesn't know me, I'm Anais. I'm in my master's here, and I'm the student's representative at the Program Coordinating Committee, known as CCP. And I just want to say that I'm pleased to represent all postgraduate students here. Also happy to be part of the committee to help our interests with Bruna Silva, who is not here right now, but she's gonna join us later. And as an invitation, the committee that is organizing the next encounter of comparative biology for the next year is inviting more people to compose the committee. So if you like to join, you can talk to Bruna or to Martin. And also, I wish this event can help all students and advisors to know better about the variety of research that are being done in our program.
Want to say something? Should I go ahead? Hello, everybody. Good to see you, all of you here. I must say that I was expecting more people here. Where are everybody? Well, I'm being involved in this comparative biology program for about 10 years now. The last 4 years I've been working with Milton on the coordination. In the next 2 years I will be leaving without the great work that Milton did, but he will help us on the side, of course. And I will have the support from Fernanda. We will work together, Fernanda Brando. And I'm sure the next 2 years will be great for all of us. We're on a stretch of time, the next 2 years, that is very optimistic, at least for me. And I hope for all of you. We had difficult times in the past. We're still feeling the repercussions of the isolation that we all faced. And doing research and working on the academic environment is collaboration, is getting together. And this opportunity here, this symposium, as was said before, is a great opportunity to exchange ideas, exchange experience, and come up with new ways of looking how things works. So it's a pleasure to be here. I wish you all a great 2 days of interaction, and I'm very excited that the comparative biology program is doing well. Okay, thank you.
Thank you, Tomás. First, I would like to highlight Anais speaking that she is calling to students to help to organize the Encontro de Biologia Comparada that will be held in the next year. We have a very good sum of money to do so. So they need people to help organizing the Encontro de Biologia Comparada. So contact Anais, contact Bruna as well as representatives. Joana, Martin is here, Martina also, and the people that is organizing this meeting, because they will need a lot of help to organize this kind of thing. So, and other thing that I would like to pay attention is the diving day. Okay, I know that the students are full of things to be done, but we had this kind of event that was held here in the Faculty of Law in Ribeirão Preto Escola de Direito de Ribeirão Preto a few years ago, and after the pandemics, it stopped to be organized. So I asked the students if you could organize the Diving Day as well, because it's important to show our program, to show the things that we have here in our faculty. I am seeing Malu, for example. Malu was of the organization of at least one or two diving days in the past. So I would like to stimulate you to resume this activity that is extremely important to our program. Okay, Malu was one of the organizers, so ask her how this kind of event was important to our city, not only our program, but our city, Ribeirão Preto. Okay, and about Tomás, all the best in your coordination. In the closing ceremony, I will highlight it again. But all the best for your coordination. That will begin in 10 days, I think. You and Professor Fernanda Brando will be the coordination of the program, and I feel that the program is in the good hands with Tomás and Fernanda heading the program. Okay, thank you very much for all of this. Well, it's time to the first speech. Yours? Okay, so I will turn the floor to Professor Tomás that will give us a lecture about our program and the future that this program will be. Okay, thank you very much, Tomás. Thank you, Daniel and Anais as well. To all of us, a good meeting and have fun. Okay.
Well, again, it's great to see you guys here. And Newton approached me about 2 weeks ago saying that he would like that I present something for you guys. And I didn't have much time to think about what I was going to do. You were very involved. Vera, hey, welcome back. I was very involved on completing one of these grant proposals to FAPESP. Many of you here experienced writing a proposal, submitting, and all the anxiety that it brings. And so 2 days ago, I started looking into this. And I decided, well, what do I want to do as an opening of this 4th symposium? And I thought of doing some kind of a review, what is a comparative biology program, and then to provoke you guys, all of us, of what we want to do for the next 2 or 4 years immediately, but in the long run as well, as to be established as a leading group on this field in Brazil and in the world as well. So here it is. It will be short, it's not that big. And the first thing I want to tell you guys is that, well, our program is not young, but it's not old as well. It started in 1997, so it only has 26 years. I'm here for 10 years now. So 26 years, for me it's not that long, but many of you guys are about that age. So what do we want with this program? Mainly, we want to increase the knowledge that we have in science. We want to understand better how the biodiversity that we have here on Earth came to be. So what's the evolution of this biodiversity? To do that, we need to understand very well the ecology that leads to this evolution. Ecology as a driving force. It's a very complex matter, but it goes hand in hand with evolution. And also, unique to our program and envy of many other programs is that we have this component that we have groups studying how this knowledge has been generated and how it changes through time, and how is the repercussion that this history has on today's landscape. Not only that, but also how we transmit this knowledge to other groups in our society. So education is a big part of our program as well. And how we do this? USP, our institution here, our department, and the Comparative Biology program has to offer the infrastructure, a friendly, we hope, environment for you guys to flourish, tools, financial resources, and our experiences to provide you guys, masters and PhD students, with this resource to become a scientist, maybe a technician, maybe an educator, and for sure some people that will have enough knowledge and experience to be decision makers in our society. So for our society, the decision making is not for our society, not for all societies. The decision making is very important. So if we are not doing these decisions, we are expected to communicate with people that do. So that's some things that we aim with our program. So, I don't know if you guys, when you thought about coming here, if you search for an advisor or if you search for the program. You're going to have a diploma, a master's diploma or a PhD diploma saying comparative biology. And what does that mean for you, right? Does anybody here came because of this program? It's called Comparative Biology. 1, 2, that's good. It's not very common. There's no name, even during the undergraduate course. It's not very well discussed. But we're not alone. We're partners, more or less, with the. Oops. There's another program in Paraná, which is also called Comparative Biology, and it started in 2005. And I put another 3 examples here. American Museum of National History. They have 3 branches: evolution, systematics, and biogeography. And this one is very interesting: grantsmanship, ethics, and communication. Texas University and University of North Carolina also have similar programs. The names change slightly. In Texas they have this phylogenetics and comparative biology, and in North Carolina they have integrative, comparative, and marine biology. It's interesting. There are some others in Europe, in North Ireland, but it's not easy to find this information. Anyway, it's not common. There's many programs called just biodiversity or just ecology or just botany and so forth. But this is our attempt to be broader enough to bring together many different fields of research that we want to create this environment where communication can be enriched by these different points of view. I put here as a reminder that we have one discipline that every one of you has to take, has to sit. And ours has 8 credits, is Fundamentals of Comparative Biology. And we're discussing this because the students came back to Milton on the last evaluation and pointed that this basic course should be broader, more englobe more areas and not focus too much on evolution. But I brought this here because the others programs also has some courses that are mandatory. So this one has evolution, systematics, and biogeography, and this more general course. But we're working on that, and hopefully this mandatory discipline will become more interesting to a broader group of people. Again. Okay, how can we describe ourselves now, right? So we do have a master's and a PhD program. For a master's program, you guys need to get 16 credits in disciplines. For a PhD, on top of that 16 from the master's, another 16. We have 50 different courses that you can choose. I was surprised, I didn't know that we have so many. And it's good, it's great. Many of them are very specific, but many of them also are broad enough to generate interest for a large variety of areas of research. Now we have 29 advisors or researchers working, advising students. We had in the past 441 theses and dissertations completed. It's a lot. In 26 years, 441. It's huge. It shows that although comparative biology is not that common as a name, it brings people from different areas, and we're doing a lot of work here. Right now we have 70 students enrolled in masters and PhD, 26 masters and 44 PhD students. And from the 70 students, 54% are women. So it's well balanced, and it's great to see that well, it's not biased, gender biased, or in a way. I was happy to see that. Okay, so that was our program, and most of our activities is directly linked to the federal system that controls and regulates the post-graduation program in Brazil, and that's CAPES. So CAPES has, I don't know, all the total of all the programs we have in Brazil, but looking specifically to our area, the biodiversity, we know that we have 142 programs. CAPES has this system of grading the programs, and with this grading, it gives a kind of a stamp of quality, not stamp of excellency, not quality, because in some ways we need to reward hard work or work that's been productive, and in another way we need to identify who we need to help more. And in a way, CAPES has this classification system and has a system to afford financial support for these programs. So this, probably you guys heard this a lot, this grades, conceito CAPES. And the maximum 7, the minimum is 3. And you can see here in the biodiversity program or area, only 9 is grade 7, 17 is grade 6, so 7, 9, 17, 26, grade 5, 48, grade 4, and so on. And we are here. We're grade 6, and I want to talk to you guys what we want to happen in the future. We want to get to this maximum position here. We want to get to grade 7, and it's a collective work, and everybody will be involved. So how does it work, right? So we have copies here that provides this platform of data gathering to do this analysis of how post-graduation programs performs in Brazil. And this is a result of the work we do here, pondered by the quality of the production that is done. There is this qualis ranking of basically scientific journals. This information, every 4 years, is compiled and sent to Sucupira, and then that goes to copies, and then we wait for we wait with our hearts on our hands to get this grading, because it translates how are we comparing to our colleagues. Here is how much money campus gets from federal sources. So this starts in 2000, this 2022, and here in 2008 or 9 we start having an increased amount of funds, but that was temporary and came down again. Hopefully this upward trend here will continue, but it depends a lot on the economy of the country. All right? So we are susceptible, very susceptible to the economy of the country on how much it goes to research. So what can we do if we want to take this last step and get into the elite of the, at least of the biodiversity programs? Every 4 years we have this evaluation, and the last evaluation 4 years ago, CAPES was, I guess it was under pressure to release how they evaluate, to disclosure the topics, all the specifics that they use to compare the programs. That was not the case before Milton became coordinator, and everybody was more or less in the dark. Only a little group of professors, usually professors on the committees, the area committees, who discuss what they thought was important and then rank the programs. But now this information is much more open, and I want to share these things with you guys, because that's what will guide us on identifying what we can improve and what we're doing good already. So CAPES separates the evaluation in three groups: the program itself, the formation of human resources, and the social impacts. And it's a mixed match, because some things here happens here as well, and vice versa. But in the program, the proposal, so our proposal, as I said before, is kind of unique. It's prized by our peers, having these three components: evolution, ecology, and history and education. It looks at the group of advisors in the program. It looks at the theses and dissertations produced by the students. On the formation, the number of students and the quality of their production, and the social impacts, what we can get to the outside of the university, and how interesting this is to the general society. It's a little bit vague, but those are points that are important. And as I was saying, the excellency it's more or less separates the post-graduation programs, because to be considered excellency, you need to be graded 6 or 7. And that means more money, more fellowships, easier way to use the money that comes from them. So from there are 4 topics that they pay attention to classify the programs into this excellency group. So if we have all of these 4 topics higher than a threshold that they established by comparing different programs, if we have 4 of them, we will get grade 7. It's automatically. If you only have 3, then we stay in grade 6. But if we lose, if we only accomplish 2 of these 4 topics, we will get grade 5. So we are very comfortable on our grading right now with 6. We almost got to 7, and what we want to do in the next 2 or 4 years, we want to look very intently in all the topics to identify what we can improve. And we can only improve if we all know what is being assessed, so we can change that. So these 4 topics are the percentage of students that are first author, and 53% or higher of the students that have a paper published is necessary. So everybody needs to publish it. It's that old saying on academia, publish or perish, of course. And there's a pressure to do that. There's lots of discussion. It's not just the number, but there's philosophical discussions of what to publish, how to publish, and what conditions to publish, and what quality we want to get. Do we want to spend a year writing a paper that nobody will read later, or should we spend 2 or 3 years working on something that will be published and then will advance science? So it's lots of discussion, but we want to make sure that we accomplish these 4 tasks. So the next one is, there is one entry there in Sucupira that we need to refer a specific set of papers that has the students and advisor as authors and co-authors on those papers. And there's some metrics here to count the points. It has to do the qualis classification of the journal where it was published. And there is this threshold here, but we need to report or indicate all these papers that both advisor and students are on the author list. There are many papers that the advisors get into as a co-author, and there's very common, mostly in ecology nowadays, that is this data papers, that is hundreds of authors. So there's several qualifiers here regarding the number of authors, the involvement, to indicate that this production, that paper published, was conceived and produced by here in this in our program. The next topic is the percent of advisors with 2 or more papers with the highest rating on the qualis scheme. So at least 71% or more of the advisors need to have at least 2 papers in these last 4 years of evaluation. And the percentage of advisors with age index higher than 10. So 86% of the advisors has to at least reach age index equal or higher than 10. You guys know what the age index is, right?
Scopus database.
Age index
Less than 10 in Scopus database.
is the Scopus database. Yeah, there are many
Not
ways to compute. So the age index is the numbers of papers you have that has been cited by at least that number. And there are different data centers that you can gather this information. We all know Google Scholar, and they find citations of your paper everywhere. So it's high, but other databases are more selective. And for example, CAPS uses the Scopus database. Anyway, there's lots of pressure on the advisors, but also the partnership between students and advisor is important. So one of the metrics involved here shows that for a master's, at least one paper needs to be published, and at least 2.5 papers per PhD students must be published. So it's not a light competition to get to the grade 7. It's a high competition, and it's a lot of effort that we put on this. And for sure, the hard work will be rewarded because being part of a program with the highest grade possible, it help us on our careers. So it's something to be proud of, but it's something that we need to work as a team to get there. And that's it. I was just bringing you guys that, yeah, we're serious. We want to get there. It's not bad to be 6 or to be grade 5. It's not. But I think we need to aim higher. We need to do our best to use the public money or the public infrastructure that we are enjoying here and return to the society. And the easiest way for us to do that is putting out all our hard work in form of publications and interaction with the society. So think about that and discuss with as many people as you can about this. Thank you. Questions? Do we have time for questions? Questions, opinions, suggestions, concerns. Open your heart. Is it scary to see this? Oh, I'm on PhD, I need to publish 2.5 papers.
Thank you, Thomas, for your lecture. Very enlightening. I'd like to know if you, as a new coordinator, do you think this is fair?
Fair.
When we talk about this kind of thing, because we know that campus is agents that is to form people to the society and also to get jobs. But you think it's fair, for example, to push the programs so far when we are talking about the excellence of programs, or do you think it's okay?
Thank you, Hilton. Well, I will be sincere here. I think it's fair. I do think it's fair. We need to, again, aim high. We need to do our best to return to the society what the society has provide us. It's not, they're not giving this to us. This air conditioning, the light we have here, the safety that we have here, the good environment that we have here. It's not a present, it's an investment. So I think it's good to stimulate groups reaching their maximum. And I also see that it's dangerous because it's a human nature condition. We compare with each other, ourselves with each other. And when we're losing the competition, we feel bad. We feel quite bad. It's not a feeling that is easy to handle. But a long time ago, I decided, well, I will not measure myself with the top people. Top people are special. They're very smart. They have a life condition that is different. They're lucky. I don't know, but you don't have to. You guys don't have to compare with your neighbor, your colleague in your lab or in the other lab. Be happy with what you do. You need to go to bed at night and rest and say, oh, I did my best. Your best, you decide what your best is. But collectively, it's good to have a goal, to aim at a goal and try to reach it. So I think it's fair. I think it's fair.
I have a suggestion. Sorry. So my suggestion might be not very welcoming by most of the people, but I'm going to suggest it. I have a concern about the qualification exam in the PhD program. Okay, I know how it goes in Brazil. People, they practice for the concurso, public concurso, and all those stuff. But my opinion from my own experience, the qualification exam for PhD students is very tiring. And at the end of the day, it's not very productive for the student. I would suggest maybe like obliging the student to publish an article in the midterm. So at least it's breaking the ice for them to publish an article, because for a student, for example, my background, I used to be an environmental consultant for half of my life. So going back to academy, it wasn't easy. So learning how to publish an article, it's a lot of work, and it needs a lot of encouragement. So if a PhD student learned to break the ice, publish a small article as a qualification exam, besides explaining his project for the qualification exam, first of all, he's going to finish his PhD with an article, which is very important for him. Second, believe me, those 4 months preparing for the qualification exam, it's very tiring. And my real opinion, it's a waste of time. It's a lot of work for non-profit at the end. So it's a suggestion for you to consider about the qualification exam. This is my opinion, and I hope that you're going to like it.
Thank you. Your opinion is not, you're not alone with that opinion. Be
Thank
sure
you.
of that.
Perfect.
We're always evaluating, and it's good that we have the students that are part of the congregation, not congregation, the CCP, that can gather. And each one of you guys needs to get to the representative and bring these provocations, right? Nothing set in stone, so it can change all the time.
Just to complete the idea, I understand and I agree with you, but at the moment, I don't think it's possible because of the rules that you have at the university, not at the program, at the faculty of here. The University of São Paulo has a regulation, and you need to have this qualification process where the students will be in front of a evaluation committee, but don't need to be explaining their work. It can be like another model that some places do that's like a class, like from a specific topic that we can choose or have a draw, something like that. But you need to have this process by the regulation of the university. So just publish won't be enough right now. We can bring that to the discussion to the higher levels of the university, but now we cannot like just finish that. We can change the idea or how the qualification can be performed by the students, but not like finish with it.
Yeah, definitely. But the point is that there's some important topic about students, post-graduation students, which is the mental health of the student. Like mental health of the student, it's as much as important as his scientific production. So this qualification process is really very tiring, a lot. I understand the university rules, it's an idea that might be applied in the future, but just I wanted to share like, yeah, we might be considering this. So we have already the post-graduation student has a lot of pressure on his shoulders. So imagine like preparing for a qualification exam, 10 lectures for a PhD student. It's a lot of work. Thank you to agree about the future planning to change it, but just I wanted to drop this idea.
Rafa, just to add a little piece of information. When we have the self-evaluation, do you remember the students when we had the self-evaluation of the program? The students, it was one of the students asked for it to be changed, the qualification exam, to another form of evaluation. And I think Thomas and I, we have talked about this, and I don't know the new coordination to evaluate this again, because I think it will be more productive to change it, okay? The problem is that when our new rules, they spent 2 years and a half to be approved. It was approved in last August, for example, and we have no time to change it. But now with a new coordination, I think it would be very good to discuss again the rules of the program and maybe probably change this subject. For example, to change the qualifications into another one that could be more productive to students and to the program. Okay, so I agree with this, and then we have talked about this, but I will let it to the new coordinator and the new heads now.
Just another word about that. We, as Professor Milton had said earlier, I'm from the chemistry department. So the past we had changed our qualification process, and it took like less than a year. So the students start to come to us and said, no, we want to have the qualification process like it was, like the old one that was talking, presenting their projects and the results and everything, because we think for us it's better than the way it's doing now. So we changed it again, and like 2.5 to 3 years later we started again with this process, but we tried. So like our experience, the students didn't like it, so they wanted to come back to the oldest or doing what's doing right now in the Comparative Biology program.
I'm going to say something that's, sorry, super funny about this one. I came from a non-democratic country, I'm from Syria. So I want to suggest a democratic process. Maybe you can consult the student, asking them their opinion. I'm an old student, I finished my PhD 2 years ago. But just like ask the students what they prefer, what's better for them, then we take the decision. Maybe what I suggested is my personal opinion, so it might not reflect all the opinion in the place, okay? So that's it, thank you.
For sure we can do a kind of referendum and ask, but students are also welcome to bring us their suggestions, right? It's very open. On this topic particularly, I guess, if I remember well, how we came to have this system was to increase the number of former students with us to get in positions at other universities, right? And that was, we didn't see, no, research is doing well, papers are being published, they're defending, but we want to get more of our students at the professor positions in other universities. So that's why this came to be in this way. Of course, for the students, much easier to, you're already working with our research and presenting that, it's much easier. And I fully agree with this health, mental health. We have that problem as well. We're overwhelmed with so many things that we have to do, so many things, right? And but I remember my days when I was a PhD student. I felt vulnerable at that time. You need to prove yourself. You don't know if you're going to make it. If all your effort to not getting a job, continue to be a student, that will pay off or not, right? So, yeah, anyway, students, this program is for everybody, it's yours. So come with your suggestions, participate, discuss all topics, from the entry exam, from the support that you're getting, from how it works at all. So bring stuff to us.
Tomas, there's one more question. We have
One more.
I want to come back to the previous topic. Like you said, we have a lot of pressure on advisors. Students have their roles too in acquiring higher grades in CAPS evaluation. But we have a lot of things that are out of their control, like citations, that we have each year less money to publish on open journals. We have each year less students in the labs, and even some research topics that require a long time to publish some articles. And I want to know if you have some news about how CAPES is dealing with this and how the program will deal with these questions that are out of let's publish more, you know.
Good. Well, we see CAPES as if it was a person, right? That we can ask and get a reply. But CAPES is formed by people like us. There's a lot of researchers or professors or students from other time in the past that are there discussing these things. So I don't think they're insensitive to these difficulties that you can identify here, but it's common to all over the world, for sure. It's an endless debate, this holy grail to find this holy grail of the balance between the enjoying the ride and getting to the end and not killing yourself on the process and returning to the society the investment that they're doing on us. They're investing on us. But yeah, it's not something to feel bad, for sure. We're a group. There are people that study, I don't know, things in rat's liver that you can have results in a week, and you can repeat your experiment 15 times, and you can publish in 3 weeks, right? Some other people do ecological multi-year variation in the climate, And it takes 10 years, right? So, as a group, we need to see as a group. Individually, again, I say strongly, none of us should feel bad and in comparison with our colleagues. We need to be aware of our own position, our own condition, and enjoy it. We need to enjoy it. It's hard to say it's that simple, just enjoy it. But yeah, don't keep comparing yourself too much. That's my opinion. Okay, we need to move.
Thank you, Thomas. It's an endless discussion, I think, and this kind of thing will be addressed and must be addressed in the next years to us, not only to achieve the CAPES objectives, but also to achieve our objectives, the objectives of the students, the professors, and our university as well. Okay, so CAPES are the guys that fund our program, so we have to pay attention about this, but we have to do our job as well. Okay, that's my message. Tomas, you have a lot of work to be done. I'm going out. Well, okay. Now we have the second supervisor talk with Professor Wilfried Klein. Please, Wilfried, let's hear. And Wilfried is from our Department of Biology, and he's been our advisor for many years. I don't know how many years, Wilfried? A lot. Wow. Okay. And he will present one of the lines of the research of our program, taking his own research as an example of what we do here in Comparative Biology program. Good, thank you very much for your presence. It was always a pleasure. Stage is yours.
Thank you, Milton. Do we really have to use the microphone?
Yeah.
It's up to you, because it fits better because, yeah.
I don't like microphones because I need 3 hands to use a laser pointer, pass the slides. So let's see. Yeah. I have a laser pointer. Thank you for the invitation. Always a pleasure to talk at home. My Comparative Biology program is my home base. That's the only program I'm working, and I'm supervising students. So when I was asked for a talk, I was thinking about a more general topic that would please more than just hardcore physiologist or morphologist that I'm known as. I had the idea to talk about oxygen, because everyone loves oxygen, everyone knows oxygen. Everyone likes it so much that you get extra dose of oxygen. You pay for it, and you have some health benefits of aging less. You can use ozone to improve your health. Various health benefits listed here. I'm not going through all of them. You can actually, I think that's a trend that has not arrived in Brazil yet, but I think in the States, you can buy a machine for just $1,100 that concentrates oxygen from the atmosphere and then feeds it through some colorful liquid to paying customers. So you go to oxygen bar and you get extra oxygen, because everyone loves oxygen. And we don't need to go back that much. Everyone knows the application of ozone up yours behind, which has no benefits whatsoever, but people were selling it as a good thing. So oxygen is a good thing according to advertisements, but if you look at other advertisements, you see, let's take a juice with a lot of antioxidants, which helps us increase our health. Pills with a similar effect. So what? Oxygen is good, oxygen is bad. What do you think? Too much antioxidants, something to fight oxygen. So what's good? What's better? Too much? More oxygen or less oxygen? Let's have a vote. Who will take more oxygen if I give it to him? No one? People are shy. So everyone takes a juice with antioxidant effects. Yeah? Okay. It's better. Because, well, based on that, you can call actually oxygen a paradoxical molecule, because if you talk to astrophysicists, if they find a planet full of oxygen, they say there is life. Because it's oxygen is associated with the presence of living organisms. But if you look back at the history of our Earth, the chemical processes, chemical origin of life on our planet happened without oxygen present. No molecular oxygen was present during the origin of life. So everyone knows aerobic metabolism produces more ATP than anaerobic metabolism. So that's a good thing. You get more energy from oxygen. But oxygen was also supposed to have enforced endosymbiosis. When, with the increasing amount of oxygen in the atmosphere some billion years ago, some organisms like tree mitochondria possibly entered a prokaryotic cell to form a eukaryotic cell. And later on, possibly also stimulated multicellularity. However, as Thomas got it right, excess of oxygen is not good. If you breathe 100% oxygen for more than 2 hours, your lungs get irritated, you feel bad, and eventually you will fall sick because of reactive oxygen species. Does everyone knows what a reactive oxygen species is? No, I don't think so. So, everyone knows this reaction, every biologist knows that. We love to learn the Krebs cycle, what happened there, electrons transported from here to there, producing ATP, transforming oxygen to water and CO2 when the substrate is glucose. However, this process here, this transformation 6 oxygen to CO2 actually involves several steps which form what are called reactive oxygen species. They have 3 electrons here in red, and these are highly reactive molecules who will cause damage to DNA, proteins, macromolecules, because they're highly reactive. These electrons are looking for other electrons to steal, and this will cause damage to other molecules. And these reactive oxygen species are linked to several diseases. And just to give you an example, someone calculated that in one single cell of a rat, we have something like 1 trillion oxygen molecules each day. And if only 2% of these oxygen molecules result in free radicals, you will have 20 billion free radicals of oxygen. So a lot of free radicals inside one cell. And these may cause up to 90,000 attacks on the DNA molecules inside the cell, and may lead to something like 10,000 DNA-based modifications per cell per day. So a lot of potential for deleterious mutation-driven genetic material. Of course, you have several mechanisms to correct for that. For example, you have specialized enzymes inside the nuclei who corrects damaged DNA. You can separate the DNA within the nucleus, so putting it farther away from the oxygen molecules that are being processed at the mitochondria. You can have specialized enzymes, superoxide dismutase catalase, that actually transform these radicals into harmless raw and molecular oxygen. And someone has suggested that sexual reproduction actually evolved because of the presence of free radicals. because the recombination of DNA material from two individuals will correct for possible modifications you can find in the DNA. So, if you look at the oxygen within a cell, it's being used at the level of the mitochondria. So this represents one cell. You have the enzymes and other stuff involved. Basically, the mitochondria processes the oxygen, and actually that happens at a very low level, 1 millimeter of mercury. That's a lot. It's a little. It's nothing. How much oxygen do we have here in this room right now? Physiology lecture, people. Everyone had physiology. Well, it's in millimeters mercury, which is a bit difficult for you, non-physiologists. But we have one atmosphere as a pressure of 760 millimeters mercury. We are at 550 meters, so a bit less. But 21% of the air inside this room is composed of oxygen, and this gives us something like 159 millimeters of oxygen in the atmosphere at sea level. And so, 1 millimeter is nothing. It's really, really low, very low amount of oxygen. But actually this is supposed to protect the mitochondria. Mitochondria cannot deal with excess, with too much oxygen. And so within the cell we have very low partial pressure for oxygen. And the mitochondria, from Comparada, let's talk about biology, comparative biology. Mitochondria are not equal. There's no one type of mitochondria. There are several different kinds of mitochondria. You even find mitochondria that don't work with oxygen, that don't have any genetic material. And just to show two large modifications that you can find within an organism, you can have tissues with very few mitochondria. All these dark spots are mitochondria. You can have tissues with a lot of mitochondria because the tissue is metabolically more active. You can have variations within the membranes, within the mitochondria. More membranes mean more enzymes associated with the Krebs cycle, so more processing of oxygen and producing of ATP. And actually, this is a very understudied topic in comparative biology, the variation of mitochondria within different animals or even plants. Daniel just left, but his lab, for example, works with mitochondria. And I found out recently that actually 3 labs here in Development work with mitochondria, mostly based, associated with to study some disease. But of course, I'm going to work with them and study mitochondria of more interesting animals. So, studying mitochondria is really interesting and fruitful. But move on. The oxygen has to get to the cells. How does this happen? We need some means by that transports the oxygen towards the cells. Who does that? Who delivers cells? Who delivers oxygen to the cells? No. Well, the lungs are far away from, far away. Middle Earth. So, blood, yes, blood, which structure delivers capillaries? Anyone think about capillaries? Capillaries are in a closed circulatory system. The end point, which with the oxygen-rich blood arrives at the tissue and will diffuse oxygen will liberate oxygen to diffuse into the cells. If you have an open circulatory system, you can call the liquid hemolymph. If you have a tracheal system, which is very common in arthropods, you don't need a circulatory system. You just deliver oxygen directly to the tissue. And the important aspect I want to talk about right now are the respiratory pigments. Respiratory pigments are carriers of first of all, capillaries. We are 19 days away from Christmas. So most of you will eat a large bird on Christmas. Am I right?
Yes.
Or people like bacalhau. Pasta and pasta. You're going to eat a large bird. Which muscle do you eat? Which part of the bird do you eat? Come on. Oh, he is special. He doesn't like the breast muscle.
No, I like, but the kids like more.
Yeah, we get the leftover. We get the leftover. So people like the breast muscle of a large bird, and this is actually a very white muscle. So we have here white muscle. And Thomas and I, we eat also the thigh, the leg. The leg is white. No, the leg is darker. It's red. It's a red muscle fiber. Because what's the difference? The birds we're eating, they're not made for flight. They're made for eating. So that the muscles that should power the rings don't have blood vessels. Very little irrigation. So the number of capillaries in this muscle is really, really low compared to a red muscle like the muscle in the thigh. Or if you would eat a hummingbird, the pectoral muscle would be really, really red, because the hummingbird obviously flies tremendously well. So you find morphological difference within the tissue based on the capillaries which are related to the amount of oxygen that are being needed within the tissue. The volume of mitochondria varies, the diffusion distance varies, and high tissues that need more oxygen have a better delivery system for the oxygen, so more capillaries. And the other thing we have to talk about are the respiratory pigments. We all know hemoglobin. Yes. We know myoglobin, which is a form of hemoglobin. We know hemocyanin. Yes. We don't know hemerythrin. But these are the three pigments, respiratory pigments, that are known. And as you can see, the metazone groups, you can find hemoglobin in plenty, hemocyanin in a few, and hemerythrin in another few. But what should call your attention? For example, hemoglobin and hemerythrin are found in worms, same groups. Arthropods have both hemoglobin and hemocyanin. So the evolution of pigments is not very simple, and actually different for hemoglobin, hemocyanin, and hemerythrin. Functionally, what the pigments do? What's the function of a respiratory pigment? I like to show that you know nothing. That you don't remember your classes. So what's the function of a respiratory pigment?
Carry gases.
What?
Carry gases on respiration.
Transport oxygen.
Transport.
No.
No.
Oxygen.
Just
oxygen. So you may remember a graph like that. That's a hemoglobin within our body, for example. At high partial pressures of oxygen, remember, cells have 1 millimeters per mercury. And here we have 100 millimeters mercury partial pressure of oxygen, and then you find that in the lungs, in our lungs. At the lungs, all the hemoglobin in the blood is being saturated with oxygen. This is a percentage of saturation of hemoglobin, 100%. And this blood, this hemoglobin, is transported by the circulatory system to the tissues, and the partial pressure in the tissues is lower. So hemoglobin will unload a part of its oxygen. More or less 25% will be unloaded and will be released to the tissues where it's being transferred and diffused to the cells and being used there. So hemoglobin is a perfect transport for oxygen. However, that's not how it started. Hemoglobin evolved a long time. Yeah? And when I mean a long time ago, I mean this. Geological history of atmospheric gases. Our atmosphere started with a huge amount of carbon dioxide, much more hydrogen than today, and a bit nitrogen, and then it changed. Carbon dioxide decreased, nitrogen increased, and oxygen started to show up in significant amounts, something like 2 billion years ago, 2.5 billion years ago. But respiratory pigments, like hemoglobin, probably evolved more than 3.5 billion years ago. Because the original function for hemoglobin was to sense oxygen, so that all the anaerobic organisms would see, oh, but there's oxygen, I will die if I stay there, let's move on. Let's move away from them. They can neutralize reactive oxygen species. All these structures that have, these pigments have a metal ion in the center, some porphyrin ring around, which is called the heme, and then some protein together. And all these metalloproteins have the capacity to neutralize, to react with oxygen. And the original function of these heme proteins were actually to protect the organism from the deleterious effects of the oxygen within the cells. The transport function would evolve only later, together with the evolution of multicellular animals. So, the pigments unload oxygen at the tissues, possibly within capillaries, but who transports the pigments through the body? It's of course some pump. We call the pump a heart, and the heart will move liquid, blood or hemolymph, through our body, through through most of the bodies, and the respiratory pigments are the ones who carry the oxygen. So, and it doesn't really matter if you have a lung, if you're a snail and have a lung, if you're a crab and have some gills. The heart will receive oxygen-rich blood from your respiratory organ and pump this oxygen-rich blood to the tissues where it will be exchanged, delivered to the tissues, and then the oxygen-poor liquid will be returned to the respiratory organ and this heart. And for example, these are two examples of open circulatory systems without capillaries. Remembering that annelid worms have closed respiratory systems, so we have capillaries there. And the pump is the dorsal vessel and the ventral contractile vessel, so it's quite diverse what you have regarding the pump. When it comes to vertebrates, we can look at fish hearts, which have several chambers in series, which are pumping the blood to the gills, and from there the blood will be delivered to the tissues. And everything changes in the vertebrates with the evolution of a pulmonary system, the lungs, which come together with a pulmonary circulation. The pulmonary circulation will use the same pump to move oxygen-rich blood and oxygen-poor blood. And starting with amphibia, starting already with the lungfish, dipnoid lungfish, amphibia, the rest you know, should know. You can have division of the heart in two atria, here as well, and then one undivided ventricle. So in these groups we have the mixture of oxygen-rich blood and oxygen-poor blood, and the animal has to somehow manage to divide these flows within their body so that oxygen-poor blood gets to the lungs or the gills or the skin, and the oxygen-rich blood gets to the tissues where it's being consumed. And this happens in all these, amphibia, reptiles. Birds and mammals have a fully separated ventricle, so 2 atria here, 2 ventricles, so no mixture of oxygen-rich and oxygen-poor blood, and we call that event shunt. The mixture of oxygen-rich and oxygen-poor blood within the heart is called shunt. And all these animals here have the ability to shunt blood from one circulation to the other. We are in the mammals at a disadvantage. We have to move the same amount of blood from towards the lungs that we move towards to the body. So we have to work. We have to move blood through the lungs, get oxygen, and this oxygen has to get has to be used. The lizard, for example, tail lizard. Tail lizards, they hibernate. Everyone sees them disappearing in April, May, September they show up. So while they're hibernating, they breathe not that much, the metabolic rate is quite low. And during that time, they are able to move the blood and it returns from the body to the heart, and they just bypass the pulmonary circulation and return this used blood again towards the body. So they don't consume the oxygen inside the lungs as fast as they would, as a mammal would, for example. So if you have a divided ventricle, you are quite fortunate of having an advantage to regarding your circulatory regulation. However, there are always exceptions. For example, if you look at the Burmese python, it has 2 atria, 1 ventricle, and this ventricle has 3 small cavities, but allows a mixture of different pulmonary and systemic circulatory blood. But if you measure the blood pressures in the pulmonary circulation and the systemic circulation, you see a clear difference in blood pressures. If the same ventricle, the same muscle would pump 2 blood streams together, the pressure should be equal. However, this snake here is able to create a systemic pressure much higher than the pulmonary pressure. This is actually a picture that you would expect from us, from a mammal. So despite an incompletely separated and divided ventricle, these snakes are able to maintain pulmonary and systemic pressures very separate, which is quite a feat for a reptile. You can find a similar thing in monitor lizards, but you don't find something like that in the turtle, for example. So quite some variation between cardiac morphology and function in reptiles. So that's the heart. And the next step is what gets oxygen into the body. Now we have lungs, single mealcom. We have lungs, gills. If there was no specialized respiratory structure, we call it cutaneous respiration, skin respiration. And we have tracheal systems which are different, but I have to mention them. As you can see, for example, there are groups which have all 3 of them. Cranials, if you just think of an amphibian, a salamander, you can find them having cutaneous respiration, they have gills and lungs. So you can have a mixture of respiratory systems in different types. And just to give you a very brief example, this is the skin of an amphibian. These are actually, no, this is not the skin, these are the capillaries irrigating the skin, which is used for respiration. And these are really densely packed blood vessels, which offer a high surface area for oxygen to diffuse from the outside directly into the skin. Something like this is a schematic representation of the skin and the blood vessels. If everyone knows gills, it doesn't matter if the gills are in a fish or an amphibian or crustacean, whatever. You have very fine lamella full of capillaries. All these dots are hemocytes, red blood cells carrying hemoglobin, able to capture oxygen, so a large surface area floating in the water, allowing the capture of oxygen from the outside. Lungs, we can have sac-like lungs. This is the lung of a lizard. And you have to breathe in, exchange your gases, and then breathe out. It's a bi-directional ventilation. You breathe in, you breathe out. Everyone could do that right now to wake up. We have to mention birds as a specialized lung because they have a unidirectional airflow within their respiratory structure. This is a very neat gas exchange part of the avian lung. And for completeness, let's show a tracheal system where the oxygen diffuses directly through a spiracle on the outer surface and then diffuses through air-filled tubes directly to the tissues. And just to make it clear, tracheal system, it takes oxygen directly to the muscle, to the tissue. There is no circulatory system involved in oxygen transport. They have a circulatory system for other functions, but not for oxygen transport. And interestingly, for example, if you just look at arachnids, its ancestor supposedly had gills, but at the base they all had lungs. You might remember book lungs, something used to breathe air, and several spiders, scorpions, for example. But during the evolution of various groups, actually the lungs got replaced by a tracheal system. And then you can ask, why this is happening in these spines. The respiratory system, the tracheal system, is much more efficient because diffusion in air happens quite faster than the circulatory system moving pigments through the body. Remembering that these invertebrates have a low pressure circulatory system, so the blood flow is relatively slow compared to a vertebral circulatory system, for example. So the trachea, they will transport the oxygen much faster, the oxygen much faster to the tissues. But still, the functional implications are quite interesting. Well, a very short snapshot of vertebral lung variety. What's that? Mammalian lung. I could show you a lung, which is basically the same. But the most interesting part in vertebrates, other animals, mammals are boring. I don't work with mammals. You can, for example, work with tail lizards. Tail lizards have a very simple lung. This is one lung which was dried and cut up in the middle. And you see just one sac, no internal divisions. These ridges here represent the respiratory epithelium. So a very, very simple lung structure for such a large lizard. The tail lizard is a large lizard and is very active. So it's actually quite surprising how it can do that with such a simple lung. If you look at other animals like a caiman or a turtle here, the lungs are more complex, have several chambers, respiratory epithelium concentrated in one area, this area is more dilated. But for example, if you look at another chelonian, a marine turtle, a sea turtle, its lungs is really, really densely packed with respiratory epithelium. Has to do with sea turtles being active, constant swimmers. They exchange gases much more, need obtain oxygen much more. In much greater number than, for example, this one here. So quite some variation within chelonians. If you have an elongated body, normally you have an elongated lung. What's this? Everyone knows this here. What's that?
Lungfish.
Which lungfish? Piramboia. Estamos aqui pelo P. Piramboia lungfish, and that's the lung of, these are the two lungs of the lungfish, and like most fishes, lungs, swim bladders are quite elongated. This one here has a lung of a rattlesnake, and as you can see, respiratory epithelium concentrated in the anterior part of the lung. This one is just an air sac, and it's blown out of proportion because to dry that lung you have to connect it to the pump, and obviously this part gets blown out more, and the living animal is never that big. So you can have quite some variety, morphological variety within the lungs of vertebrates. And what can you do? the first thing you do, if you know nothing, you study morphology. Morphology is the base for everything. So get to know the morphology of your animal, of your structure. So you can take a iguana, lizard, and then look at the morphological, the macromorphology of the lungs, and then apply microscopic techniques to study the fine structure of these lungs. If you have a lot of time, you can do that, for example, with the lungfish, where you can ask, lungfish have lungs, they have gills, yes, they are fish, they have gills, and they have a skin. So which of these structures is more efficient for respiration? So, and if you apply morphological techniques, you can count surface area, diffusion distance, and then you can determine how important is this structure to obtain oxygen or to release CO2. And interestingly, based on the morphological estimates, the lungs are responsible for 99% of the oxygen consumption, the uptake of oxygen, and the elimination of CO2. So, despite being a fish, a lungfish, its gills are totally useless for gas exchange. They might have other functions, but for gas exchange they don't. They're not good. And actually, more gas exchange happens through the skin. CO2 is easily lost through the skin, for example. The oxygen also can be taken up through the skin. So, if you have time and money, you do a lot of stereological estimations of respiratory gas exchange surfaces. If you want to do something with the elongated snake lungs we had in the lab, Barbara did this for her monograph. And actually she did look at the distribution of the respiratory epithelial angulation of the heart. The position of the heart in snakes varies a lot, which might have to do something with the snake being arboreal, going into trees or just living on the ground. And she found that in the rattlesnakes, for example, respiratory epithelium are always anterior to the heart. No, the head of the snake is here, to be clear. And in the boar, for example, the boar has two lungs, still. This was only one lung. The respiratory epithelium is distributed close to the heart and posterior to the heart. So if you make a comparative analysis, you can try to understand why this is the case. If you're a lot into physics, you can actually model the airflow within the lung. If you have a complex lung, like a multi-chamber lung in a turtle, you can put some airflow flow meters inside the lung surgically and then simulate breathing, and you can try to understand how the airflow is being generated within the lungs during expiration, inspiration, and see how actually how well mixed is the air within the lungs. It's functionally important because these lungs are really huge compared to the alveoli in our lungs. So if the air stays in one place, the distance of the diffusion distance for the oxygen to the capillary is getting larger and larger. So knowing how the air flows through the lungs is quite important. Something I work quite a lot is breathing mechanics. So you have a ventilator here, a pump, which simulates volume of different volumes and frequencies. You can see 120 ventilations per minute, and this is connected through some cannulas to a rat, and you see the animal is breathing. No, it's being ventilated because it's dead. And what you can do is you measure the changes in pressure that come with the changes in volume, and then you can calculate how easy it is to ventilate these lungs. I did this a lot in reptiles, because reptiles are more important than mammals, of course. For example, you can ask how this, the changing position in caimans, affects breathing mechanics. And because this one is resting at the surface, there's less water pressing on the body cavity compared to this one. This one, these were juvenile alligators we used, some 100 grams or so. And then you have a hydrostatic column, as the lungs would be here. So we have a difference of 4 centimeters of water pressure acting on the body wall, which changes breathing mechanics. What else did we do? Tail lizards. Tail lizards have their lungs and they breathe. So this did this work? And then you can ask tail lizards to reproduce. So a female one, can carry, a female tail lizard can carry a lot of eggs inside the body cavity. So this will impact the volume of the lungs. Here's the cavity for the lungs. And all these eggs will impact lung ventilation, breathing mechanics. So we measure that. I measured the same thing in the turtle, and that's maybe it's lighter because the animal's living in a box. So how do you ventilate lungs in the box? Of course, with only more spaces associated to the legs. And we studied breathing mechanics in these turtles. Paulo, who did his master in the program, studied jabutis, red-footed tortoise. Baby ones, large ones, to see if the growth of these animals affects breathing mechanics. We're currently working on that documentation. Back to the lungfish. You can study how does the air get in and out of the lung, in and out of the animal. Because it's an air-breathing fish, he has to go to the surface, gulp some air, and then move the air to the lungs. So you can put some cannula into the buccal cavity, into the lungs, and you see how the pressure changes when the animal is ventilating, how the oxygen concentration in the lung changes over time. So you keep the animal in a tank. He has to breathe into a funnel, and they take all the measurements. Quite fun, actually. So that's some things you can do with the respiratory system. How much time do I have? Another 10 minutes? Okay. So if we look at all the transport cascade, we start out at high levels of oxygen in the atmosphere, and then it decreases constantly to 1 millimeters per mercury in the respiratory in the cells. And then, if you don't want to, if you want to know how the animal as a whole is infected, just this cardiorespiratory system when it's challenged, you, for example, can put the tail lizard on a treadmill, and then put a mask on it, and then have it run on a treadmill, different velocities. This one here is to measure just ventilation, so you just need the animal. This box is here, so that animal is not escaping. You can challenge it by temperature. This is a lady from Saudi Arabia. I did some work on it several years ago, and you can see how the ventilation happens at 25 degrees compared to 35 degrees. So temperature for ectothermic animals is obviously a really important factor. I don't need to talk about climate change, physiology, so on. I actually can't tell you if an animal species can deal with climate change or not. You can challenge the animal by feeding it. I could challenge you going to the nearest churrascaria, and everyone eats a kilo of meat. Everyone will do nothing at the Invicta tonight. Will just lay flat because it has to digest. And the tail is similar. You can put the tail in a box, and you measure the amount of oxygen and CO2 inside the box. Over time, this one experiment was carried out for 2 weeks or so. So the animal has a resting metabolic rate during the night, which he increases during the day, so there are no variation. And here at 0 hours, I gave the animal some meat, which he happily ate, and then afterwards he showed an increase in metabolic rate due to the ingestion of meat, of the meal. Ectothermic animals, they have a very low basic metabolic rate, and when once they ingest an animal, some food, they will increase it for various amounts of time, which depends on temperature, meal size, and other stuff. So, talking a bit about Ray's PhD project. He is working with these birds. People who are close to my lab will have heard them singing a lot. And actually, the most interesting part of this bird is their hearts are really small. That the size of their hearts are like a crocodile. Okay, this one is the heart of a chicken and other birds. And as you can see, these birds fly, but they don't like to fly. If they can run a race, they do that much more often than to fly. They can fly for 2 minutes, and that's it. And the idea is to study the respiratory, cardiorespiratory physiology of this animal, and we measure basic metabolic rate. This video showed Ray visiting a colleague in Bolivia, and then running a different species on the treadmill. So the idea is to challenge the animal while running on the treadmill, and then measure oxygen consumption while doing that. And actually, we also work on the morphology of the respiratory system. So Ray will work on this part, on the heart as well. And on the mitochondria, actually, we have a collaboration with Danielle to measure the function of the mitochondria in the species. And to understand how the transport of oxygen is affected in this it works in this animal, and how it's actually limiting its exercise capacity for flying. Other things you can do is to look at the various oxygen concentrations in the environment. I think everyone knows that. In water, oxygen concentration can vary from a lot to nothing. Especially if you look at Amazon, or other tropical water bodies of water, they can become anoxic during the day, for example, or during the night, some part of the day. The air also shows variations in oxygen. Just to remind you, going up a mountain, you will have less oxygen available for you to breathe. So, for example, at the top of Mount Everest, you should just have 1/3 of the oxygen available that we have now here. So, you can challenge the animals doing that. To other species of turtles, normal oxygen levels are here, and then we challenge them with 9, 7, 5, and 3% of oxygen, and say this is ventilation, all the amount of air they were moving over time, and actually they don't care if there was no oxygen around. They care if there was a lot of CO2 in the atmosphere. So CO2, 3% for some animals will increase ventilation a lot, which is typical for terrestrial air-breathing animals. So we can do that. What else? Evolution. Biologists love evolution. So you can look how these lungs are distributed over phylogeny. This one is a crocodilian lung, this one is a sphenolon lung, monitor lizard lung, and then these are other lizards. And you can ask, how did all of this actually happen to occur? And Barbara's master project, just like this, just that. She is reviewing the literature regarding lung structure, basically of reptiles, and then she's doing several phylogenetic analysis to understand what kind of lung was at the base of squamate lizards, and how actually how every taxon itself evolved. Additionally, she's not here right now because she's at the University of Florida in the United States, where she's analyzing CT images of several lizards that were scanned there. For example, this alligator lizards has a lung, three different stages of the respiratory system to understand how the lungs are being formed during development. Well, Diego did something similar in tadpoles. He was a master PhD student here. But to wrap it up, one big question people like to ask is evolution of endothermy. Why is endothermy present in birds, mammals, and then interestingly in several isolated taxa like some lasmograngs, several fish. And again, what's here? The tail is it? The tail is a fantastic animal. During springtime, it actually is an endothermic animal. So this graph is relatively easy. The black is the temperature of the animal in January, April, June, October. Yellow is the temperature, the air temperature, and green is the temperature of the underground dam they live in during the night. And as you can see, in January, the temperature of the animal is just a bit higher than the bird where they're living. During the day, increases body temperature, and then it falls down. The same in April and June. June is hibernating, that doesn't get out when it falls. But during reproductive season, actually, the animal is able to maintain a 6-degree difference between the environment and its body. So it's a faculty, it's an endothermic animal during reproductive season. No one knows yet how this endothermy is produced. It could be just muscular contractions, could be some hormonal regulation. People in Jabochkabal are studying that. So just to give you an image, this one is a tail within a bird during the reproductive season. It's much more warmer than the outside. And this is the two-tailed lizards here, inside the body having the same temperature, inside the bird having the same temperature as the environment. So the multiverse. The multiverse of opportunities. But if you just look at the transport of oxygen from the outside to the inside, it involves several levels, several systems. And you can study all of this using different techniques, whatever you want. And the same applies, obviously. The same system is used to eliminate CO2, so oxygen gets in, CO2 gets out. And several things, all of these topics, evolution, development, all the different levels of organization, physiology, morphology, behavior is a very important part to study the function of an animal. And obviously environment, so ecology is also important. So you can do a lot of different things. And it's quite a challenge to do everything alone, so you need collaborations with other people. And I think you've noticed, you can do it here. Teiu, lang, piramboia, are very common animals here. Teiu is really easy to get and just go outside, grab one. And they are really good animals because they're relatively large, easy to maintain, very resilient when to perform experiments on them. So it's actually quite a lot of opportunities if you're interested in organismal function when it comes, in this case, to the transfer of oxygen from the outside to the inside. And not even mentioned the nervous system or central regulatory mechanism, which is a whole different world. I don't know. So I hope you enjoyed this rapid flight through the animal kingdom. Some students and collaborations that were involved in all this study, and of course, funding agencies. And with that, I'm happy to take your questions.
There was a time in the evolutionary history that we had high oxygen levels, for example in the Paleozoic, and also we had large organisms. I see papers that say that body size is correlated with oxygen, high oxygen levels, but they don't describe how this actually works. Do you know how this works?
Large body size being associated with high oxygen levels. So I skipped the slide, I have no time. But as you can see, the largest amount of oxygen in our atmosphere, something like 35%, was something like 300 million years ago during the Carboniferous. Then it dropped during the, actually it started dropping, dropped to its lowest point during the Triassic, down to something like 12%, and then started to rise up again. So, well, probably Max can talk more about that. Some lineages, you have, for example, giant insects, arthropods, during the Carboniferous. Everyone knows that giant libella. But actually, the libella is not a good, very good example, because libellas are what? Long tubes, and the diameter of the libella is just of this one was just 3 centimeters. So the diffusion distance from the outside to the inside is just less, 1.5 centimeters. Nowadays you have beetles that the largest beetle is something like 15 centimeters. It's a very massive insect. So this one has a much more challenge to obtain oxygen than the giant libella, and it was flying around during the Carboniferous. But I don't know the paper you're referring to, but maybe the animals were larger during this time and then dropped in body size during when it's entering the Mesozoic area. But the basic respiratory system for birds and the dinosaurs evolved during the lowest oxygen concentrations present. The origin of dinosaurs somewhere in the Triassic, and the bird-like respiratory system, which was supposed to be have existed in dinosaurs, they permitted to allow them to fly, obviously, and also to achieve the large body size of sauropods and other large reptiles.
Thanks, Wilfred. I have a question on the chambers in the birds and mammals. You say that 4 chambers in the heart of mammals and birds. And the blood doesn't shunt. Is this correct?
That's correct, yeah.
For the rest, I don't know the correct word, but for the rest of the reptiles, for example, we don't have this kind of arrange in the heart with 4 chambers and not shunt. Yes. Is this a limitation to the reptiles when you talk about homeothermy and high performance or not? Because you have 2 different origins of 4 chambers with no shunting blood, for example. Is it a kind of limitation to reptile-like animals, for example, to fly or to perform better, to run or something like that?
Well, a limitation depends on the definition of limitation. Flying animals, flying reptiles, dinosaurs they probably have a 4-chambered heart, like birds and mammals do, to generate enough blood pressure and to pump the oxygen-rich blood efficiently towards all the tissues. But for these animals here, you have actually crocodilia a bit. They have a 4-chambered heart, but they have the ability to shunt the blood through secondary evolution, which is the foramen or appendix, which is some other stuff. But for example, within the lizards, we have monitor lizards, which are really highly aerobic animals. They can run for quite a long amount of time. And they can generate much higher blood pressures in the systemic circulation than the pulmonary circulation. So they can functionally separate blood flows, like a mammal and a bird. But it's temporary during high activity phases. When they're resting, they're shunting blood, and that actually saves them energy during resting periods or when they're hibernating or whatsoever. So actually it's an advantage that they're able to do both for some of the species. But most lizards and turtles, they just have to shunt the blood most of the time. But some few exceptions can do it. Okay. More questions? Lars.
Hi, Wilfred. When you talked about respiratory pigments, I think hemoglobin, and the origin was something I wasn't expecting, that it's actually to not repel, but no, to offer oxygen. And this is a theory, it's something that is well proved. I didn't heard of it, I never heard of it, so I want to know how that was established. I don't know.
Yeah, well, hemoglobin is present in several metazoan groups, but also present in bacteria, archaea, maybe even plant. So it's a very ubiquitous, very present molecule. And actually, for example, if you look at the structure of the chlorophyll, which has a porphyrin ring, just as a metal ion is magnesium. So it's a different metal ion, but it's also reactive to oxygen in the same manner. And all these are very early evolutions within the Earth system. It's quite established that hemoglobin started out as something different as a transport molecule, just more as a defense mechanism against oxygen present in the atmosphere. Hemocyanin is a different story because it evolved from a different origin within arthropod groups. But hemoglobin is mesomorphic for all the metazoan organisms present in various groups of bacteria. So I'm quite sure. Another question? No? Oh, thank you.
Okay, last one.
In German, please.
I don't mind. Thank you for the talk. I was thinking, imagine a person that was born in a high altitude and a person that was born in a sea level. Are they really different in terms of oxygen consumption or respiratory system? Because we have a colleague in the lab and he's a really good runner. So I was thinking it might be different. I don't know.
So the effects of altitude on the development of human phenotypes is being studied in people in Tibet and people in the Andes. And well, I don't know the genetic studies, it's not something I read. But from a functional point of view, people have analyzed, for example, the performance of the Sherpas carrying stuff in the mountain in Himalaya compared to normal people. And actually Sherpas have a way, a behavior to carry the load in a very slow way. They walk slowly, and if they have to breathe, they stop and replenish the oxygen. So physiologically, if you raise animals under hypoxic conditions, they tend to be of smaller body size, but maintain the size of the lungs. So proportionally, they have larger lungs. And of course, what you refer to, they have a higher number of red blood cells. So if you live at high altitude, erythropoietin, a hormone, increases your blood cell production, so you have more blood cells carrying oxygen. So the blood is more capable of transporting oxygen, yes. But the lungs morphologically, no. Okay? Thank you.
Thank you. And just to make it clear, all the students that Wilfred showed, they are from our program, is it correct?
Yeah.
Yeah, okay. All the students, Bárbara,
Raí, Tabata,
Tabata, Paulo,
Pedro was on the slide.
Diego,
Pedro.
all four. Thank you very much.
Hello everyone. Today I will present some results of my 6-month internship in Germany. My advisor here in Brazil, the Dr. Enichimatsu, my internship advisor was the Dr. Gabriel de Souza Ferreira. The title of this presentation is the Young Callus, exploring interspecific variations in Callus arenocensis using CT scan. So in this first slide, I put a summary that what I will present today. So I will start talking about the student group and why I chose Germany to do my internship, the goals and the resultant experience that I had in Germany. So the student group, my student group is the Testudines. The Testudines has two main lineages. Biologia, or side-neck turtles, and crocodiles, or high-neck turtles. The main difference between these two lineages is the form of the reticular neck, with biologials attacked laterally, and crocodiles attacked anteroposteriorly. The Testudines is a very successful group. They inhabit in a lot of environments like marine, freshwater, and terrestrial. And currently we know 357 species described. And inside the Pleurodilus contains the genus that I studied, the focus of my work, is the genus Callus. This genus is a very specific, they have specific features. And they have four species, Callus umbeatus and Callus arenocensis for the extant one, and Callus columbiana and Callus lewisi for the fossils. The main difference between these two species, the extant species, is the shape of the carapace. Callus umbeatus has the quadrate shape, and Callus arenocensis has the oval shape, like this follows. And the features, they have some features very specific, like a flattened skull, reduced eye, and some fringes in the face, and a tubular nose. So in this study, I made some CT scans of some species to try to find some interspecific variations in some features. Now, why I chose Germany? I put two main reasons here. The first one is the location. In Germany, they have good museums with a lot of specimen fossils. I put three examples here, like the University of Tübingen Museum, Zekenberg Natural History Museum, and Paleontological Museum in Munich. And the second reason was my advisor, is the professor, the Dr. Gabriel. I chose him because he's a specialist in turtles, And in the past, he's already studied the same genus than I, the genus Calus. So, in my opinion, was a good choice. Now the goals. I put 3 main goals here. The first one is the CT scan. So we took 5 specimens from the Dezembro Museum. So we conduct CT scans. The second goal is the segmentation. So after the CT scans, we segmentation all the specimens using the software Amira and Dragonfly. But mainly the Amira because it's more easy to use. Here the logos. And the last one is the photogrammetry. This step don't have relation with the first and the second because it's with another genus, with the genus Stupendennis. Because in my last visit in Acre, I had contact with some vertebrae and I took some photos in different views. So when I showed to Gabriel, he gave me the idea to do this photogrammetry. He bought the software and we did this photogrammetry. The results now. Here is some photos of the segmentation. So in the left we have a shell. In A we have a dorsal view with the carapace. In B the ventral view with plastron. And in C a set of cervical vertebrae. In the left we have the 2 views of the skull. In A, an entire skull, and B, half of the skull. In lateral view, in C, an entire skull in dorsal view, and D, in dorsal view, a half skull in dorsal view. Here is some appendicular bones. So we have a hand with the carpals, metacarpals, and phalanges, radial ulna, scapula, humerus, and coracoid. Here and with the left, we have a foot with all bones, tarsals, metatarsals, and phalanges too. Tibia and fibula, a humerus, and a pelvic girdle. And to finish the results, some of the 3D models. So in the left, we have the more complete vertebra. So we will describe this specimen, and to complete we will do a revision of 4 specimens already described. But this specimen was described in Portuguese, so we think it's a good thing to describe in English all these specimens together. Other specimens. With these CT scans, we will do here in Brazil because to do these segmentations took me a lot of time in German, so I need to finish here in Brazil. But we will describe these 5 specimens that we scanned, and we will do a complete description to create an anatomical atlas of the Calus adenocensis that can be used to future comparison. Now my experiences in German. First I put the scientific collections in museums. In Zekenberg I went to take the specimens to get the segmentation. It's a very good museum. I put some photos here. So we have a raptor with some eggs, turtles, and a monkey in very elevation. The famous Archaeopteryx. Forgot the name of this. I forgot the name, sorry. And this, I don't know the name. It's very complicated to say. In the second place, I put the University of Turin Museum. That was where I stayed all my time there. I put a ichthyosaur, a mosasaur, and some plants. To finish the museum, I put some pictures of the Paleontology Museum in Munich. And now, I had the opportunity to participate in a conference that took place in Spain, more specifically in Salvador City. This conference was organized by the European Association of Vertebrate Paleontologists, and I presented a poster. And in this conference, I had the opportunity to see another works and talking with other researchers. Was a very good experience. And here the city that I stayed all my time is Tübingen. It's a very small city, but it's the same as an old city. So there are a lot of beautiful places to visit. In the first photo, we have a castle, and here is a bridge, postcode of the city. And to finish, I put some personal experiences some years, because the Germans know how to do some good years. And thank you for all the university that made this work continuous. Thank you. Question?
Thank you. One question about not the society, but the work environment. You work in Germany, for example. Do they have different kind of behavior in a lab, for example, when compared in Brazil?
I don't know. Here in Brazil, we have more contact with people in my lab in Germany. Each people work solid. They don't like to divide the opinions. But I don't know. In my lab it was like this, but I don't know now if it's like this. But here in Brazil is more. Yeah. No offense.
Always depends on the supervisor.
That's why your face is here.
Do you have a fun story to share with us? Like
Story?
something, some trouble or something
Yeah, I had one.
strange?
One night I was eating Burger King. It was night, and a wasp took me in the neck, and I came loud and just put my hand in my neck, and sure it was a bit his laugh a lot.
That was very fun. No more questions? No?
Thank you very much.
Hello, so my name is Danilo, and I went to Australia, to Melbourne, to Monash University, and I will talk a little bit more about the background that made me want to go there. And well, my research there is, the name is Relationship between Head Development and Diet in Lizards, and I use the Tropidurus catalensis lizard, which is the calum that we all know. Okay, so my master's is ideally divided into 3 chapters. The first one, I look at interspecific diet evolution in the family Tropiduridae, which is the family of those lizards. The second one, I focused on the interspecific difference in the diet and their relationship with head morphology. And those species, they vary a lot regarding diet. I mean, they vary evolutionarily if you look at the phylogeny, the terminal branches, and they vary interspecific. For example, two populations can have very different diets. And so this led us to the third chapter, which is called Individual Phenotypic Plasticity, Individual Phenotypic Establishment due to Diet. And this chapter here is the one which led me to Australia. So, I don't know if you all know what's phenotypic plasticity, but it's basically the ability of the same phenotype or kind of similar phenotypes to express different, genotype to express different phenotypes. So this is one example of a research in our lab here in Brazil. And those fishes are all from the same species and populations, but they were fed with different, they were fed with diets in different places in the cage. So they developed entirely different head shapes. And we have very few examples of phenotypic plasticity related to diet in lizards. We have one paper from 2016 in which the authors captured adult lizards of this Eolianus species, and they fed the lizards with a diet that is rich in plants and a diet that's rich in animals. And they noticed some morphological and microbial changes in the gut of those lizards in, I think it was a month. And in Tropicuridae, the family that I studied, they have very different head shapes. And I discovered, because of my first chapter, that we have evolutive associations between the amount of plants they consume and also the amount of hard prey they consume and the shape of their heads. So we started to think that maybe some species might exhibit phenotypic plasticity in their heads. So we had two aims in this study. First is to test if the lizards of the species Tropicuridae catalamensis can express phenotypic plasticity in their heads according to diet. But also we would like to test if those morphological changes could have functional implications for feeding behavior. If, for example, animals that fed on hard prey would be best in processing the hard prey. So I have some videos here. I don't know if but okay, no. Wow. Sorry. Okay, sorry. Those are some very cute videos of lizards eating things, but you will not be able to see it. But anyway, everything started with field work here in Brazil. So those are the females of the calango lizards, and they are graphed. You can see that they look like golf balls. And we captured a bunch of those graphed females because we needed to obtain their eggs and incubate those eggs in laboratories so we could have small baby lizards to test. And then I divided the lizards into 3 groups. The first one I fed with cockroaches, with a soft prey, really cockroaches are soft. And the other group was eating the majority of hard prey, which is jataí bees. And the third group were fed with mainly plants, which were small pieces of apples. And I managed to keep them in laboratory for 4 months. Then I euthanized the lizards, and we took CT scan images here in USP Ribeirão Preto. I was about to explain to you what a CT scan is, but you already know because of Programa. So my goal was to have those finite element models that we can use to do finite element analysis. This analysis is like building a physical material of something that you want to test in a physical scenario, but you do on the computer instead of, for example, printing in a 3D sketch or something like that. So this is one example of a paper from my supervisor in Australia, in which they basically simulated the school of this monkey during mastigation, and with this technique you can kind of invent physical scenarios for the structures that you build. So you can, for example, see how the stresses and the forces are applied to the bones or another structure that you want to see during any given physical activity. So we have one example, insofar as I know in the literature, of someone that used this technique in animals that were raised in captivity. My supervisor in Australia hated this paper, but I didn't know at the time. But anyway, those lab rats were fed with 2 different diets, and they used the technique to see that, for example, the animals that were fed with the hard diet suffered less stress during mastigation. So this is like the goal of the research. So to start, then I went to Australia because I needed to learn how to construct the models, because, to be honest, it's kind of a tricky thing to do, because it's very easy to commit mistakes, and you have a lot of details, and you have a lot of things that you can do wrong, and you will have wrong results. So it's kind of stressful, but I went to Australia to learn how to construct those models with my supervisor, who is a leading developer of a lot of those methods. So I took the CT scan images of the lizards, and first I needed to segment the skulls, that is, to make a 3D model of the bones. Then I needed to use mainly the jaw, because according to my supervisor, the jaw is more likely to have suffered modifications, because you don't have so many functions as the cranium. For example, the jaw doesn't have many sensorial functions. So this is a jaw of 4 months old calango, which is very cool because they have a lot of those rows of teeth like small sharks. And after that, we needed to insert the information about the muscles of the animals, because if I want to simulate a realistic feeding scenario, I want to simulate a bite. So we need to have information about the insertion of the muscles and the vectors that connect to the muscles. And I was very lucky to have Juliana here, my colleague, who could dissect some lizards in our collection. And she kind of drawed the muscle insertions in a cranium of an adult of the same species. It's a very high level and highly detailed information. And I modeled the surfaces of the muscles in this scope. Those are the centroids, because using those centroids, we can kind of, you can see, but we can kind of calculate the vectors of the muscles to make kind of virtual muscles of the animals. And you need to have a lot of data. For example, the muscle force of some related species, you need to have the information about the EMG and the area of the muscles and a lot of things. So you can create this 3D muscle. And in the end, you have a model that looks like this. So this is the jaw with the vectors of the forces applied. So those yellow arrows are kind of, you can imagine it, the virtual forces pulling the bone, because those are the muscle insertions. And you use kind of constraints to say, for example, okay, so this teeth and this teeth, they cannot move because the animal would be biting something, and you have a virtual model of the animal doing something. So the results are very preliminary. Actually, I will have a meeting with my supervisor in the end of this week. But we have those images of the force distribution in the jaws, and those are the 3 experimental groups. And this is a lizard that I fixated as soon as he was born. He's like the standard, let's say, without any influence of diet. Those are those colors represent the stresses, like how much the bone suffer forces under a bite, and these gray areas are because of the constraints. It's an artifact of the simulation, unfortunately, because where you push the constraint, the force appears way higher. And as I said, it's very preliminary, but we can kind of see that, for example, the neonates, they suffer way more stress in the bones, which makes sense because they are way smaller, they have less bone, they have less teeth, they have way less everything. And between the 3 diet groups, it's hard to point to you, but the lizards that ate the soft material, they appear to suffer less stress. That means they may have more resistant bones because of the diet. The animals that ate hard prey, they seem to have more fragile bones, kind of. And the plant group is super weird because they have like this bimodal shape and this strange line here. My supervisor says that this line is super weird, so we are still kind of analyzing, but those are the preliminary results. And I will just thank my 2 supervisors, and Juliana, and the funding of FFCLRP, the Monash University.
Could you observe differences in the manipulation of the food in the mouth?
So if I could show you the videos, I would say yes, because the lizards that were, when they were eating the bees, they seemed to not masticate in the normal way, but for example, the cockroaches, they would just bite and swallow. And the bees, I could see that they keep doing this movement of from one side to the other several times. And the apples, not so much, but more than the cockroaches. But I didn't do an image analysis because I don't know how to. I have some videos, but that's it.
Sorry. I know you won't answer the question, but would you expect differences in muscle mass manipulating the jaws?
Maybe. So when you do the surfaces of the muscles and the whole model, you need to escalate the forces according to the animal. And one thing that I still don't know what to think of is that the animals that fed only on cockroaches, they were bigger than the other groups for some reason. I could see, like, when I was looking to the models, I could almost predict the group of the animal without seeing. So the muscles are surely different, but I don't know how to analyze that. I only know how to scale the forces around.
Which forces did you calculate with finite elements analysis? Which force? Which force?
Oh, the strain, the von Mises, and there is one analysis that we are still deciding if we want to do. I mean, I did, but we don't know if we think it's appropriate. So you kind of cut your model in 10 slices, and you have, it's called moments, you have the vector of the forces inside the bones, and you have one of those moments in each of those 10 points, and you can kind of overlap the points to see if the distribution of the forces are different in those points.
Yeah, but did you calculate the, I don't know, the pullback bites, something like that?
Yes, I only simulated the bite. I don't think the pullback makes sense because the things that they each are very small. They don't need to cut the food into smaller pieces. So I simulated the food, like a bite in the very tip of the bones.
About the muscles, it was clear to see the insertions.
Huh?
It was clear to see the insertions of the muscles in the bones.
I don't know. Okay, so with Juliana images, I could kind of draw, but the babies were kind of different from the parents. But you can see, for example, so like here, we have a very clear concave shape in the bones that we could use to draw the surfaces. Here is the same. Some muscles were harder, for example, this one. But the thing that my supervisor said is that since we are comparing groups, it's more important to be consistent between all the models than to use the exactly perfect surface for each muscle.
I'm just asking because in fishes it's very common use some techniques to prepare the specimens to see the muscles and the bones. And I don't know if you are still doing this work and you can apply something for your research.
My colleagues.
You can have, I guarantee you can have beautiful images about muscles.
Yeah, so my colleagues did this. They used the PP something, the color because to see the muscles in the CT images, you need to use a specific technique to have the right contrast. And we used that, but the time necessary to make the 3D muscles
I'm
was so
not
high.
talking about the CTs, I'm talking about the specimens in alcohol. The preserved specimens that you have, I'm talking about that. You can use like some methods to put the bones in red and you can see the muscles very clear.
Yeah, we did this, but the problem is that the time necessary to do the segmentation, this already takes a very long time, and for segmenting each muscle manually is kind of impossible. So until now, we didn't use for anything, but if you have any suggestion.
Okay, we can talk about
more.
You can dissect the individuals. You don't need to do this on CT scan. You can do it manually. So you can see the muscles.
Oh, you mean measure the muscles?
Yes. You dissect the individual. You can color bones, like Mando said. Color bones, color cartilage, and then you dissect. So you just use to cut the skin, and then you can take off each muscle that you want to analyze, and you can take off all the muscles so you can measure the volume easily.
Okay, let's stop.
That's okay. I actually didn't understand the patterns of phase one analysis.
Those are not the final results.
Yeah.
Okay.
Just you said that the soft individuals with soft food have stronger bones.
Yeah, in the sense of, for example, do you see the red colors? Those are the highest forces that we registered in the bones, the highest stresses, kind of. So the red parts of the bones are kind of bending more in a way. We can discuss about it. So, for example, the neonate, do you see how red it is? Because it's a newborn, so the bones are kind of weaker. if you see the animal that fed on soft material, it has way less areas in the jaw that are red. It's basically that. And here, in the plant, for example, look at the size of this area in comparison with this. This is way more fragmented. This is only orange. Those are the differences in the distribution of the forces.
It's because I thought that I would see the opposite.
Yeah, me too.
Better. And the force that used in the model is the same for all three.
It's not the same because it's scaled for the size of the model. So it's per surface area. It's scaled. Technically, size should not be a factor here if we made everything correctly.
I thought it would be the opposite.
Me too.
So I don't understand why.
Me too. Me neither. It was the complete opposite of what I were.
Do you have any idea till now?
I can't speculate. Do you want me to speculate? Yeah. So, for example, there is a theory about phenotypic plasticity that when animals are exhibiting phenotypic plasticity during the ontogeny, they grow slower. We have empirical data with snakes. So maybe those individuals are kind of the normal for the species, and those two groups are exhibiting phenotypic plasticity, therefore growing slower and having bones that are more typical of younger individuals. It's just speculation, it's something that I think.
One more question.
One more question. Why Australia?
Because my supervisor, she's very good with the technique. She's from a lab that makes the methods of research. So, for example, she is now developing ways to implement the viscosity between different structures. So it's between all the labs in the world that do FEA, it's a very good one. But our lab already has a partnership with her lab because of the FABLES, which is a scholarship from FAPESP and Monash University. So we already knew her, and this made things easier in terms of diplomacy.
Thank you, Donato. Thank you, Danilo. And could you imagine they are Master in Science students? A couple of years it would be impossible to send students of Master in Science degree going abroad in USP, in the campus, in USP. Things are much more easier now than 10 years ago or 15 years ago. You're lucky